Hello and welcome back. The topic for this video, we're going to talk a little bit more about DNA structure. We're also going to talk a little bit more about the nitrogenous bases. Before I get into that though, I want to talk a little bit about the central dogma that is associated with molecular biology. Okay? The central dogma is something that the majority of scientists hold to be true. Okay? I will tell you with the central dogma, because this is biology, there are some exceptions in there. But what we've got as far as the flow of genetic information, we've got DNA. That DNA can undergo replication to make more DNA. And we'll talk about that in a later video. Okay? That DNA can be converted into RNA, ribonucleic acid, by the process of transcription, which we also haven't talked about yet, but we will. RNA then is converted to protein by the process of translation. Okay? Generally, when we're talking eukaryotic cells, this is how information is processed within the cell. The DNA can replicate itself. DNA can give rise to RNA. RNA can give rise to proteins. Where we have exceptions with that are going to be with viruses. So again, this is how viruses have been important in our understanding in how each of these molecules work. Okay? Viruses can have as their genetic information either DNA or RNA. So viruses can take RNA, convert it to DNA, then follow our central dogma of converting RNA into protein. Okay? So that's kind of the exception. But what we're talking about in this class, we're talking about eukaryotic cells. So I want you to focus on the central dogma and the flow of genetic information here being DNA can replicate itself, DNA can give rise to RNA, RNA can give rise to protein. Okay? That's going to become important when we talk further about transcription and translation. In the next set of videos, I'll talk about how DNA actually replicates. Okay? Before we do that, we're going to talk a little bit about some of the similarities and differences with RNA and DNA. So DNA is going to be double-stranded. It's going to use the bases adenine, thymine, cytosine, and guanine. All right? And the sugar is going to be deoxyribose. Okay? The other thing that's going to be unique about DNA, it's going to exist as a double helix. RNA primarily is going to be single-stranded, although there are some types of RNAs that will kind of fold on each other. It's not double-stranded and does not form a helix like DNA will. All right. Our bases with RNA are going to be adenine, uracil, cytosine, and guanine. So we have different bases. Our sugar is going to be ribose instead of deoxyribose. Okay? So those are kind of some of the differences between DNA and RNA. Some of the things that are the same between them. If you notice when I listed the bases, 3 of our bases are the same. So adenine, guanine, and cytosine are all found in both RNA and DNA. Where we see a difference is going to be with thymine. Okay, so that's going to be important. All right, both RNA and DNA exist as monomers, as individual nucleotides. Those nucleotides have a phosphate group, a sugar, and a nitrogenous base. And I've drawn that on previous videos, so I'm not going to draw that again here. You've got some more diagrams in your PowerPoints if you want to take a look at those. All right. I want to talk a little bit more about the bases, the nitrogenous bases. And if you notice over here, I've drawn them. Okay. Again, don't memorize the overall chemical structure of each of the 4 bases. I'm not going to ask you to draw the 4 bases, but what I will ask you to be able to distinguish, I want you to be able to distinguish between the bases that are considered pyrimidines and the bases that are considered purines. So I have conveniently color-coded this, okay? And our pyrimidine are going to be the ones in blue. So thymine and cytosine are going to be our pyrimidines, okay? In green, we've got our purines, okay? Our purines are adenine and guanine, all right? And if you have some of the supplemental material, there's some shortcuts to kind of remember purines versus pyrimidines. Purines, a lot of times the way people remember that, the thing that's unique with the purines, if you notice, each of those has a double ring, okay? And if you think of a double ring, you have 2 rings when 2 people get married, okay? So one way that students sometimes remember the purines are as pure as gold, okay? So our purines are going to be our adenine and our guanine. Pyrimidines then are going to be a single ring structure, our thymine and cytosine, all right? And I put up the structure because that's going to become important when we put together our DNA double helix. Because in order to have our DNA double helix spaced properly, and so it forms that nice spiral, okay? What you have with base pairing in the DNA double helix, you are always going to pair a purine with a pyrimidine, okay? And what that's going to do is that's going to keep the space between our 2 strands of DNA equal. Because if you notice, if we pair adenine and thymine, we've got 3 rings. If we pair guanine and cytosine, we've got 3 rings. That'll keep the space in between our 2 strands of DNA equal, okay? So you're always going to pair a purine with a pyrimidine. And in the case of base pairing, there is a scientist who figured this out, and the name of that scientist was Chargaff. So we have Chargaff's rules for base pairing. So with the rules of base pairing, adenine always pairs with thymine, cytosine always pairs with guanine, okay? Now, in terms of that, if you may have noticed when I wrote those up, I put 2 lines between adenine and thymine and 3 lines between cytosine and guanine, all right? If you didn't notice that, I want you to notice that now, okay? Because that's going to become important in how these particular molecules base pair, okay? Because what happens with adenine and thymine, we have 2 hydrogen bonds that hold those bases together, okay? Cytosine and guanine are going to have 3 hydrogen bonds that are going to hold those molecules together. And I've got a diagram in your PowerPoints that shows you where that hydrogen bonding is happening between which molecules in those two different nucleotides. I'm not so concerned that you memorize that part of it, but I do want you to know that you've got 2 hydrogen bonds that form between adenine and thymine, 3 hydrogen bonds between cytosine and guanine, okay? What that means molecularly, okay, is it's going to take more energy to melt a triple hydrogen bond than it does to melt 2 hydrogen bonds. Okay? So I want you to keep that in mind. But what's also important about Chargaff's rules is the fact that adenine always pairs with thymine and cytosine always pairs with guanine means you can figure out how much of each nucleotide you have if you know one of them, okay? Because you know the amount of adenine you have has to equal the amount of guanine. So if the adenine in your DNA molecule is 20%, that means, because A always pairs with T, you've got to have 20% of your DNA molecule be thymine. All right? So hopefully that makes sense, right? So that leaves us with now guanine and cytosine. So 20 was plenty. We've got 40% of our DNA molecule that's adenine and thymine. That means 60% of our molecule has to go between cytosine and guanine. Okay? So that tells us that cytosine is going to equal, I can do math, I promise I can, 30%. Guanine also going to equal 30%. Okay? That gives us 60% of our DNA molecule in cytosine and guanine, 100%, because those are the 4 bases you're going to have in DNA, are accounted for. All right? So the base pairing is going to be important with the DNA double helix, and we're going to talk a little bit more about how base pairing is important when we get to DNA replication as well. Because if you know the order of the nucleotides on one strand, you know what the order of the nucleotides has to be on the other strand. Okay? Hopefully that makes sense. If you have any questions, please let me know.